Hi everyone, welcome back to my educational channel on biology. Teacher Janet here. In this video, we're going to discuss the production of insulin by transgenic bacteria using recombinant DNA technology. The learning standards are as follows. After this lesson, we should be able to explain the application of genetic engineering in the production of insulin by the transgenic bacteria. And number two, we must be able to apply the same principles of genetic engineering to explain new examples of genetic engineering based on a diagram that's given. This came out in SPM 2021 and 2022. So this second comment is my own. And since this topic is very important for SPM, first of all, we have to understand, the principles of genetic engineering and how to use the different terms like host organism, recombinant plasmid, and so forth, as we'll study afterwards. And also try to obtain this book of the past year biology SPM questions, all right, for 2021-22 from the online shops. And try to question yourself after you have studied the principles of genetic engineering here in this video. So here's our concept map again on chapter 13, genetic technology. We already discussed genetic engineering, the meaning and the applications. We have discussed this first part, the genetically modified organisms, right? And we have learned some of the terms. So now we are at here. We're going to discuss the production of insulin by genetically modified bacteria. And in the next video, we'll discuss genetically modified foods. Production of insulin by genetic engineering. Here's the introduction. Now, what is insulin? Insulin is an important hormone produced by the pancreas, and its function is to control the blood glucose level of a person. So when the blood glucose level is high, for example, after a meal, the pancreas will secrete insulin. Okay, this is in a normal person. Pancreas will secrete insulin to convert the excess blood glucose into glycogen in the liver, right? But for patients that who suffer from diabetes mellitus, they do not produce enough insulin to convert all the excess blood glucose into glycogen. So they suffer from consistently high blood glucose levels. Now, in the long run, if the blood glucose level is always high, it can cause serious effects like blood vision, nerve damage, excessive thirst, slow healing of wounds, and kidney damage. Okay, so these patients need insulin injections daily to reduce their blood glucose levels. Now, previously, this insulin was extracted from the pancreases of animals like cattle and pigs to be used to be injected into these diabetic patients. But nowadays, human insulin is commercially produced by transgenic bacteria, such as those from the species Escherichia coli, okay, as seen here. Now, this transgenic bacteria is produced through genetic engineering using recombinant DNA technology. Okay, so these bacteria are special bacteria that are not the normal type because they have received a human insulin gene that is transferred into their cells. Okay, and this will enable them to produce the human insulin, although they are bacteria. Okay, so as for this species, which is commonly used to produce the transgenic bacteria, okay, is E. coli. We sometimes call them E. coli. Now, you can see that it's rod shaped, and there are two forms. The motile form that can move have flagella, which are hair-like projections that stick out from the body or cells. Okay, and there's the non-motile form that do not have flagella, such as seen here. These do not move much, and so there are two forms because sometimes we see them with the flagella and some without. Okay, now other successes or products of genetic engineering are hepatitis B vaccine, blood clotting factor, and growth hormone. Okay, so for example, we have already studied about the blood clotting factor that's produced by the transgenic goat, right? So the transgenic goat is a product of genetic engineering, okay? And growth hormone is usually produced by transgenic bacteria. So before we discuss the production of insulin by bacteria, let's look at the 5 basic steps that are used in recombinant DNA technology. So this is just one method of recombinant DNA technology. Okay, there are other ways that the recombinant DNA technology is used. In this method, a plasmid is involved to act as a vector. Okay, and the plasmid is a circular DNA from a bacterial cell or bacterium. Okay, let's start. Now, firstly, we have to take out or isolate or extract the DNA from the donor, such as humans, or it could be other organisms. So the DNA with the gene that we want to use, right? Or the desired gene. Okay, so locate first of all the gene in the chromosomes. Which chromosome has that gene? For example, the human insulin gene that codes for the production of insulin. Okay, it is on chromosome number 11 in the human cell. Okay, so we locate that and take out the long DNA strand on chromosome number 11. And after that, we have to cut out the desired gene called the gene of interest or the required gene from the DNA of the donor, right, from the humans. And we use restriction enzymes to do it, which is a special type of enzyme. Okay, so the gene is a segment, just a shorter segment of the long DNA strand here. So we cut it out specifically with restriction enzymes. Okay, and after that, let's look at the other part, which is the bacterium. So this bacterium, we're going to take out the circular DNA from it, which is called the plasmid. And the plasmid is going to act as the vector to carry this desired gene. So we have to cut open the plasmid using restriction enzymes too. So now you have two components, the cut plasmid and the desired gene. So the plasmid is cut so that there is a space for the gene to be inserted inside here. Okay. So next, to join these two parts together, we have to use another enzyme called DNA ligase enzyme. Okay, so this special enzyme, DNA ligase enzyme, helps to combine or join the two strands of DNA together. The one from humans, okay, the desired gene from humans, and the plasmid. So now this new plasmid with a new gene in it is called a recombinant plasmid, because now there's new combinations of genes in it, right? So it's sometimes just called recombinant DNA, okay? Recombinant DNA or recombinant plasmid. Now this plasmid has to be inserted into a host organism for the gene to express itself. So insert the recombinant DNA or plasmid into a host organism, such as a bacterial cell or bacterium, or it could be a plant or other organisms. Okay, and so the genetically modified organism is produced. The host organism is called a genetically modified organism. Okay, it's a special organism that has new combinations of genes. It's sometimes called a transgenic organism because it carries a foreign gene from another species, such as humans. Now, then we clone the host organism to produce many genetically identical copies of it, right? So that each of the cells will have this new recombinant plasmid. And the gene in this recombinant plasmid, the foreign gene here, like the gene for production of insulin or the gene for production of growth hormone, it will direct, this gene will direct the synthesis of the protein that is required. Okay. discuss the production of insulin by genetic engineering. Firstly, we need the cell that is going to donate the gene of interest. Okay, so the donor cell is the human cell, and in the nucleus there are many chromosomes. So we have to find the right chromosome, which is actually chromosome number 11, that has the gene for production of insulin, the gene that has the instructions or the code for the production of insulin. Okay, so take out the whole DNA strand with the gene in it first from the nucleus of the human cell. Then cut out the human insulin gene from the DNA, the long strand of DNA, with restriction enzymes, right? So this is just a symbol to show the restriction enzymes. So after that, we have the gene for insulin, production of insulin. It's a human insulin gene, right? As you can see here, the DNA is found inside the gene. So the gene is a segment of the DNA. Next, we have to get a bacterial cell that contains a plasmid. So a plasmid is actually just a circular DNA in bacteria, and it can be used as the cloning vector. A vector means something to carry the gene of interest or the gene that we desire, which is actually the human insulin gene. So we're going to put the human insulin gene into the plasmid. Okay, the scientists will put the human insulin gene here into the plasmid. But first, we have to cut the plasmid. Cut open the plasmid so that there's space to put in the human insulin gene. Okay, so again, cut the plasmid, which is actually a DNA strand, cut it open with restriction enzymes. Next, you have now got the two parts, right? The human insulin gene and the plasmid from the bacteria, which has been cut open. So we are going to put in the human insulin gene, insert the human insulin gene into the plasmid, the cut plasmid. So insert insulin gene into the plasmid, right? The yellow part here is the insulin gene. Insert into the plasmid by using another type of enzyme called DNA ligase enzyme, which will join the two strands together at the blue parts here. Okay, so in the end, this whole structure is called a recombinant plasmid because we have recombined the DNA from two species into the plasmid. Okay, we have the bacterial DNA in the plasmid and then the human insulin gene or the human DNA in the plasmid. So now we have this recombinant plasmid with the human insulin gene inserted into it. So next we get a bacterium or bacterial cell, for example the E. coli bacteria, and insert the recombinant plasmid into the bacterium, right? So what is produced is a transgenic bacterium which has the recombinant plasmid. It is called a host bacterium because it contains this recombinant plasmid, and it can produce whatever the human gene directs it to do. Okay, for example, with this recombinant plasmid that has the human insulin gene, this transgenic bacteria will be able to produce insulin. All right, so transgenic bacterium has recombinant DNA or plasmid with genes from two different species. So it's called transgenic bacterium. Transfer the gene from one species to the other. Okay, so it has a human gene transferred into it. Then it's allowed to undergo binary fission, which is a form of asexual reproduction. And the new daughter cells produced are all clones that are genetically identical to the first transgenic bacterium here. Okay, so all of them have the human insulin gene in them, which will direct the synthesis of insulin in the cells. Okay, so you see all the insulin produced here, the red dots. Now, so bacterial clones multiply and produce the insulin, and next the insulin is extracted from the transgenic bacteria and then purified and packaged. Then it can be used to treat patients who who are suffering from diabetes mellitus. So here are some of the terms that we have discussed in genetic engineering. So the first one is restriction enzyme. It is an enzyme that cuts the DNA molecules at specific regions to isolate a gene from a DNA strand, or to cut open the plasmid. Number 2 is another enzyme called DNA ligase. So it is an enzyme that facilitates the joining together of DNA strands. That means it helps to join the DNA strands from two different species, as an example, together by catalyzing the formation of a chemical bond, and it will form the recombinant DNA. It will help the scientists to form the recombinant DNA. Number 3 is the word vector. So vector is something, a structure that carries something. So vector in this case is a DNA molecule such as the bacterial plasmid that is used to carry the DNA or gene of interest or the desired gene, which in our case here was the human insulin gene, the desired gene. Okay, so it's carried in the bacterial plasmid. So that bacterial plasmid is called a vector. Then we have the word recombinant DNA or recombinant plasmid. It is the DNA or plasmid produced artificially, as shown here, by combining DNA from different organisms. That's why it's called recombinant. We have recombined the DNA, all right, from two different organisms compared to the original strand of DNA. So it is inserted into a host organism, such as another bacteria, a bacterial cell, or it could be into any other organism, to produce new combinations of genetic material in that host organism, so that the host organism will have some specific beneficial trait. So here are the notes concerning the production of insulin by genetic engineering, which we discussed just now. So the notes are quite similar to the notes in the previous slides, okay, showing the animation of how insulin is produced. So you can pause this video and then copy down the notes if you like. Look at this hot question. What are the advantages of using bacteria compared to other organisms to produce insulin? 3 marks. So this is a hot question. Firstly, bacteria reproduce quickly by asexual reproduction through binary fission. So all the daughter cells or clones produced through asexual reproduction are genetically identical to the parent cell, right? And the daughter cells all have the recombinant plasmid containing the human insulin gene. So this enables all the bacterial cells to produce large amounts of insulin at a high rate. Now, secondly, bacteria are easy to handle and to culture. And so production of insulin is cost effective, meaning it can produce good results. All right, this production can produce good results without costing a lot. Okay, the next hot question, the last hot question. What are the advantages of genetically engineered insulin? 4 marks. So the first advantage is that large quantities of insulin can be produced quickly as the bacteria reproduce quickly through binary fission. So the production of insulin will be faster if it's the genetically engineered insulin compared to production of insulin from the animals, right? Now, B, human insulin is more effective at treating diabetes than animal insulin. Why? Because human insulin is comes from the same species as humans. So it is less likely to cause immune reaction or rejection or allergic reaction in humans. And there are no ethical issues concerning the killing of animals because no animals were killed to produce this insulin. Lastly, there is no risk of transferring infections from animals to humans in this case, because animals are not involved at all.